Hello, everyone, and welcome to today's webinar. My name is Carmen Guguta, your host from Technobis Crystallization Systems, and today we are bringing to you the webinar on enabling small-scale analysis during crystallization screening through a single platform. Today's webinar is brought to you by our Senior Application Specialist, Dr. Thomas Kendall. You'll hear about him shortly, but seeing who is attending today, some of you might have been already in contact with Thomas before. Before we start, a little bit of information about this webinar. The webinar is being recorded and it will be made available to you tomorrow or the day after tomorrow. You will receive an email where you would be able to rewatch the recording. And also on our website, you'll have the slides of this webinar. If you have more questions after the webinar, please do write to us at info@crystallizationsystems.com or to my email address, carmen.guguta@technobis.com, or to Thomas directly, thomas.kendall@technobis.com. I think he will also share his email address shortly with you. If you have questions during the webinar, you can also write to them in the chat function of this webinar. Just before we start, for those that do not know Technobis Crystallization Systems, we are a Dutch company located in the north of the Netherlands. We are a world-leading technology provider for solid-state research, process development, and formulation. In our portfolio, we have 3 products currently. Stay tuned, soon is coming a 4th product. Currently, Crystal Breeder, Crystal 16, and the Crystalline instruments, more than 700 instruments sold worldwide. Some of you might already know these instruments, the Crystal 16 or the Crystalline, or both of them, or even the Crystal Breeder. Although today's webinar is focusing on the Crystalline and some of the new features that and software packages that they are now available for those users that they have this latest version. I would like to still remember to you that the Crystal 16 is your instrument for solubility, metastable zone width, solid state screening, any type of solid state screening, salt polymorphism, co-crystallization, amorphosolid dispersions, even, you name it. The Crystal Breeder is the gadget type of instrument that comes with its gadgets, as I call them, the setups for controlled evaporation, so connected to a vacuum pump, so you can control really the evaporation in each one of the reactors, or vapor diffusion. It's really an instrument that enables you to grow single crystals. You can also do solubility type of screening, but more like qualitative type of screening to know if something is crystalline or soluble or not. Same also from a solid state screening point of view. And we came to our webinar today, the product that we are presenting today, the Crystalline. The Crystalline instrument is working at a slightly larger scale, up to 5 milliliter, working volume between 2.5 and 5 milliliter, 8 independent reactors. And each one of the reactors comes with transmissivity, particle view imaging, so cameras of different magnification, so you can see almost close to the nanoscale, but also bigger particles, depending on what are the needs. Or if you have a Raman spectrometer in your lab and you would like to connect it to this Crystalline instrument, it is possible. And then you perform inline type of experiments. You see exactly what is happening during that you are performing an experiment. So you can follow polymorphic transformations, for example, with Raman, or even some chemical transformations. With the particle view imaging, you will hear much more about it today from Thomas and its new packages that it comes with. You can visualize, besides the fact that you can visualize your experiment, you can see exactly what is happening and when it's happening, at what temperature, what conditions, so you don't have to stay next to the instrument. But also you have information about particle size distribution, particle count, shape, and so on. Color cameras are also available. So if you had a color compound, you work with color compounds or formulations, dark ones, usually the very brown, dark solutions that are always a challenge. Now with this technology, it is possible to see more into this type of experiments. And I will go further. Of course, if you would like to know more after the webinar about one of these instruments, you can contact us directly through the website or crystallizationsystems.com, or write to us at info@crystallizationsystems.com. And as I mentioned to you, our speaker today, Dr. Thomas Kendall, he's currently our senior application specialist since a few years back already. And but a bit about Thomas: he has more than 9 years of experience in solid form and crystallization development in the pharmaceutical sector. He earned his PhD in chemical engineering at CMAC at University of Strathclyde on investigating nucleation mechanisms in pharma systems. Previously, before he moved to Technobis, he worked for Pharmorphix, which was part of Johnson Matthey at that time, and I think now is Veranova or so it's called, as a solid form scientist investigating customer compounds in pharma and agro sector. Has an extensive experience in polymer salt co-crystal screening, and as well in crystallization development using process analytical technologies, experiments, and modeling software up to kilogram scale. Thomas has also authored many articles, patents, webinars, book chapter on solid form and crystallization development. And this is about Thomas. Thomas, I am making you a presenter, and you can share your slides, I think. And thank you, Thomas, for doing this webinar today and sharing with our attendees today some of the features of the new software, of the new Crystalline that, well, not that new anymore. We just brought on the market end of last year. And some of the achievements with this instrument. And if I'm not wrong, at the end of the, at some point towards the end of the presentation, you will also give us a live demo, right? With a glimpse to the software and what one can do.
Yes, that's correct. So I wanted to show the possibilities of the software, and I felt rather than a few slides or video, it would be better to show in person live what can be done with the software.
Super. Thank you very much, Thomas. The floor is yours.
Okay. So thank you, Carmen, for the introduction, and thank you, everyone, for attending and taking the time to be here. So as Carmen said, what I'm going to be discussing today is how you can use the crystalline to conduct small-scale crystallization experiments and capture the important information that will help you design a process through the single platform. So crystallization is a commonly used process in many industries, pharmaceuticals, agrochemicals, and fine chemicals such as pigments. But within the pharma sector, it's primarily used because the majority of molecules are issued in solid form. This is due to their ease of application, so you can easily take a tablet every day rather than an injectable. as well as the high patient compliance. But it's a very complex process that has a whole bunch of factors that can influence it. So this can include the chemistry, so the actual compound itself, or any impurities present, the solvent systems that might be involved in the crystallization, the thermodynamics of stuff, things such as temperatures and pressures and other phases, maybe oils or gums can also play a role, as well as the process. So the scale of the operation itself can have a massive impact on the outcome of a crystallization. So because of this level of complexity, it requires a lot of experiments in order to understand the landscape of the crystallization. And through this, you need to conduct a large number of experiments, which need to be accurate, robust, as well as cheap to perform, because if you're producing several samples, you need to be able to do it in a way you can just use easy consumables, such as vials. But it also can be quite a material intense process. So if you want to use some of the latest technology from PAT tools, which are fantastic in helping you to develop crystallization processes, which typically is done at the 100 milliliter scale, that can be quite a material consuming process. So if you have something which is a very nice system, has only one polymorph, is a very simple cooling crystallization, which is an ideal project, you could be doing 6 experiments, which isn't consuming a lot of material. But if you encounter something we call a challenging project, which has maybe multiple solid films, you're wanting to isolate a metastable form, their potential oils or gums, as well as looking to do an anti-solvent crystallization, and the material is really highly soluble, you could be looking to do 32 experiments. So, up to 640 grams if you're doing that in the 100 milliliter scale. But if you're doing in the 2.5 milliliter scale, which is what the Crystalline can do these analytics at, you could be using as little as 16 grams, so a saving of 624 grams of compound, which could be a whole small-scale manufacturing campaign. So, as I said, a lot of the molecules in pharma is used in the solid form. So crystallization is primarily used as a way of getting it into the solid form. But it's also used as a way of purification, so being able to remove impurities that might not be able to be removed from the chemistry. But it's also a way of applying solid form control, so producing the preferred solid form every time by controlling the nucleation point through seeding, as well as influencing the particle size and shape, so changing the cooling profiles and anti-solvent profiles, or using the addition of additives in order to control the particle size and shape. And ultimately, a controlled crystallization is necessary for manufacturing and legal filing. So you need to be able to produce documents stating how your material at the end of the day is going to be exactly as you claim it will be, in the right form, shape, and size. Because if you ever take a tablet, that tablet hasn't been tested on, because usually the tests would be destructive. So you have to have confidence in the crystallization that the material at the end of the day will be what you claim it's going to be. So there's several important factors that go into crystallization development, and the 4 that I've highlighted I consider to be the most important. Maybe the 5th, which I haven't included here, is purity, but that's usually done offline, is the solubility. So understanding the solubility landscape is very important within crystallization. It's the foundation, and I'll touch on that. The polymorphism, so ensuring you're getting a phase pure material. And finally, particle size and shape are important for the efficiency of downstream processes. So first, we'll be starting with solubility. So solubility is the amount of material you can get that you can dissolve in a solid into a liquid at given conditions. So at a given temperature and pressure, you'll be able to get a certain amount of material to dissolve. If you then go to another temperature and pressure, a different amount of solid will then be able to dissolve in that same solvent. And it's an important factor, and I describe it as the foundation of crystallization development, because anything else you put on top of that is going to be dependent on how well you understand the solubility. If you don't have a strong understanding of the solubility landscape, either in one solvent or in the range of solvents, or solvent anti-solvent ratios, you're going to start running into problems as the margins forever, as you go up in scale, become even more difficult to control. And by collecting solubility curves and understanding the solubility in the metastable, you can create phase diagrams which give you an understanding of your process immediately. So in blue we have the solubility curve, and in red we have the metastable curve. So from this you can draw your phase diagrams, looking at your different phases. So you have your solution phase, where everything is in solution; suspension phases, where you have a mixture of solids and liquids; and then your metastable phase, where you have solution, but something that would crystallize if a nucleation event occurs. You also are able to define a seeding window, so looking at a window which you would be able to then successfully seed your crystallization without it spontaneously crystallizing. And you can also do some very preliminary calculations to give you a basic theoretical yield, looking at a start temperature and a starting concentration, and then looking at the final concentration at your end temperature. And using this, you could create an approximate theoretical yield. And that can inform you if you need to start looking at the additions of anti-solvents in order to give you a boost in final yield. And with the understanding of your solubility curve and metastable limit, you're able then to design cooling profiles which would give you the ability to maximize your yield, as well as giving you this control over the size distribution you're wanting. So in cooling profile 1, you cool quite deep into the metastable zone before adding seeds, and then cooling close to the metastable limit will give you lots of smaller particles and not really be a growth-driven process, because you're more likely to have nucleation by hugging the metastable limit. Phase 3 would give you larger crystals, so this is where you're seeding close to the solubility, so your seed sustains, and then cooling very, very closely to the solubility curve, and that allows you to maximize growth and try to minimize the amount of secondary nucleation, and that can limit the amount of small particles and focus on a growth-driven process. So collecting solubility, there's 3 main ways you can do this. And the first is equilibrium concentration. So this is where you slurry material in a vial for up to 72 hours or a week. You then take an aliquot and you extract the liquor, so you filter off the solids. You then dilute this and typically run this on a HPLC column. It's a very accurate method for measuring solubility, but it's also very time-consuming, requires many lab hours and the scientists to create a calibration method, dilutions, and you only really get one concentration from this method at a time. Another method is the solvent addition. This is where you start with solids and then you add liquid in aliquots and then make an observation if it dissolves. Again, this is a very time-consuming process. The scientist has to be present at all times in order to keep adding additional aliquots and making observations. And then you only really get one temperature when you're doing it this method. And the third is the temperature variation method, which is utilized by both the Crystal 16 and Crystalline instruments. So in this case, we start with different concentrations of solid and liquid, and then we heat them up, and we then detect when they dissolve, so we get a clear solution, and when we're able to see when they crystallize, and when we get crystals appearing, we can then say this is our cloud point or our metastable zone limit. So using this method, it's very easy to get solubility curves in as little as 4 hours, and the whole process is automated, where you set up the program, press play, and then you come in the next day and the results are there for you. So typically with the Crystal 16, this is done by the LED transmissivity sensor, where we have LED lights going through each sample, so we can detect how much light when the sample dissolves. The Crystalline also has this ability, but we find that it's not as optimized for the Crystal 16. So we can use other techniques for using the crystalline, as well as if you have colored samples which can absorb on the 623 nanometer wavelength, such as blues or greens or dark reds, then we can use the camera technology and the particle detection in order to gain our solubility and metastable points. So this is a sample of nickel sulfate, but we're also able to reproduce this on cobalt sulfate and copper sulfate. So in light blue is the transmissivity, and you can't really see it on the graph because this is a green sample, so it's absorbed all the LED light, so there's no measurement. So in the past, this would have limited our ability to measure the solubility. But with the crystalline, we're able to measure this using the number of particles detected. And this is the dark blue line. So this is the number of particles that have been detected by the software. So as we heat up, we can start to see the number of crystals being detected goes down and dissolves, and then becomes flat, and we have just background noise in the sample. So these can just be small impurities or artifacts that are left over. And then we go up as we start to cool down, and we get an increase in the number of particles detected as crystallization occurs. And then we can see the number of crystals throughout the rest of the experiment. So using this, we're able to say, the software, we're able to set limits and say the number of particles detected at a given time. If it's above this number, it is our cloud point, so when it is crystallized. And if it's below this number, we can say this is when our clear point has occurred. And then once you've assigned this pin, so the pin will be automatically placed, you can then fine tune this to find the last image where crystals were present. So this is on the colored camera here and on the black and white. And then for the cloud point, you can reposition the pin to find when the first crystals appear. And that can be stated as your point of nucleation. So using this, we're able to get solubility and metastable zone limit curves for the nickel sulfate, copper sulfate, and cobalt sulfates. But what we noticed actually with the nickel sulfate is an odd behavior with the metastable curve. We would not expect it to be this like linear relationship. We would expect maybe to follow parallel to the solubility curve. And using the crystalline, we're able to actually look at the images to determine what was going on. So we observed two different morphologies, the first being the needle morphology, shown here in the green on the left. And then on the right, we saw these block-like morphologies of actually quite interesting, like multiple blocks sticking out of the central. And what we, after doing some reading and some additional papers, we found that there's nickel sulfate forms several different types of hydrates. So the heptahydrate forms needles at low temperatures, which is when the lower concentration would crystallize, and this gives the needles. And then the alpha nickel sulfate hexahydrate forms blocks and would nucleate at higher temperatures, and this is then what we got with the higher concentration. So this kind of ties into my next point of looking at, which is polymorphism. So polymorphism is obviously very important because you want to have something that's phase pure in order to make sure when it goes into the patient, it does exactly what we're expecting. So what is polymorphism? Very basic explanation. It's just a different way molecules can pack within the crystal lattice in order to give you different effects. So amorphous, which is no long-range order, but is very short. This is very highly soluble material. We have different polymorphs of the same material where they're just packed the molecules differently. And if we incorporate secondary molecules such as water or solvents, we get solvates and hydrates. Or if it's ionic bonding, we get salts. Or if it's hydrogen bonding, it's co-crystals. And why it matters, even though there is quite a small difference just in the way you're packing them, is these can have vastly different properties. So the first group can be described as biological factors, so how well the drug performs when it enters the body. And this is related to solubility, dissolution rate, and bioavailability. And then economic factors, so how easy is the molecule to handle and process further down the line, and these related to morphology, density, flowability, filterability. And the famous case of ritonavir, I'll keep it quite short because it's been covered quite extensively. This is where an unknown secondary form nucleated in a liquid formulation in a semi-solid capsule and then contaminated all subsequent batches. And this, in the end of the day, cost Abbott in 1996 about $250 million, which in today's money is about $750 million. So there are several ways you can determine the polymorph of a material. So this can be done using DSC by looking at the differences in melting points. Primarily is XRPD or single crystal X-ray diffraction. However, a lot of this is all done offline measurements. So the one way which you can do polymorphic analysis in situ and in real time is using Raman spectroscopy. And the crystalline can be seamlessly integrated with a whole range of different Raman spectrometers. So shown here is the version 1 of the crystalline connected with the Tornado Raman. And essentially all it is their probe comes into here instead of the camera. So on the day we build the bracket and they just insert the probe on the day, and then you're able to track in real time, non-invasively, the polymorphic transformation or polymorphs present in your sample. So an example of this is we were able to conduct using mefenamic acid, which is a non-steroidal anti-inflammatory, which has 3 polymorphs, where form 1 is the most stable and can be formed in the majority of solvents in lots of cases. But we were able to get a vial of mefenamic acid form 2, which is produced in DMF. And once isolated, we then took a sample of this, suspended it in ethanol, and stirred at 25 degrees C, taking Raman spectra throughout the experiment. And over the course of 10 minutes, we were able to track the COO stretch decrease over time until we had full conversion from the form 3, which is indicated, form 2, sorry, which is indicated by the COO stretch, down into it disappeared, in which case we only got the form 1 being present. And then finally, I want to just discuss particle shape and particle size distribution. So the effect of particle size distribution is primarily how it influences the processes downstream of the crystallization. So if you have, ideally you want a very narrow, tight, controlled particle size distribution. But if you have a broad one, it affects multiple steps. So if you have a filtration with a broad one, and I have an example later, it can give very different performances on how long it takes to filter and isolate. If you have a very narrow particle size of the desired particle size, then you can in fact remove additional processes such as milling in order to give, which can be saving time and money in a manufacturing. Mixing, ideally you want to have an equal mix between your API and your excipients. But if you have a broad particle size distribution, you're going to get localized high concentrations of your API, and therefore you don't have a uniform homogeneous mixture. Tableting, different particle sizes have different compressions and breakage properties. If you have maybe quite a lot of large particles with some smaller fines, you can have the particle disintegrate quite easily. And further, for a formulation, if you have a broad particle size distribution, it can make it difficult to standardize the amount of excipients and binders, because again, you can have localized high amounts of API if you have large crystals, and then in one part, and then small crystals in another part. One thing I always found interesting is if you ever see tablets with a small indentation in the middle of them, this is because they're designed to be, you can have these broken because the mixing and the homogeneity of the tablet is complete. So there's an equal amount of API on the right as there is on the left of the tablet break. And that's ideal because then you have complete control of the formulation. So Particle shape and size can have a massive impact on filtration performance. So on the left is an example where you have filtration of needles, and they like to form this bird's nest sort of superstructure with lots of channels and pockets, which can then trap your solvent. So there's very little solvent channels. There's no direct way for your solvent to be drained off these. It will get trapped in a lot of these pockets. Those pockets will then likely contain impurities and trap residual solvent, so they'll need to have additional time for drying to remove that excess solvent. And this can be, a slow filtration, which is maybe 1 hour at gram scale, but could be days on plant. So it's like the manufacturing cost then starts to ramp up. In second is the filtration of a polydispersed cube. So this is where you might not have a tight control of the particle size distribution. So here, there's okay packing. There's nice, you can see by the height of the filter cake is now reduced. There is some solvent channels where the solvent would be able to drain. So there would be an okay and a reasonable drainage isolation of your solid from the liquor. But there are still solvent pockets where the solvent might get trapped. And this then, during the drying stage, could result in inclusion of impurities. So something on this could be 30 minutes on a 1 gram scale, could be hours again on a plant. And as I said, there's a very high chance for impurity inclusion in these pockets. And then finally is filtration of a monodispersed cube. So this is where you have high control of your particle size distribution. Here we have good packing, so the height of the filter cake is even lower. And there's good solvent channels, so we're able to have a fast extraction of our liquor with very little solvent pockets, so we're not really trapping any impurities potentially dissolved in our mother liquor. And this can be a 1 milliliter, 1 min at 1 gram scale, which can be like an hour on a plant, so a very fast filtration time. And this can be, again, as described, this can be tied to your polymorphism, and we can see that the transformation of one form to another. So this is carbamazepine, which is a very commonly investigated compound, which has a Raman and a morphological transformation for polymorphism. So we can have form 2, which are these needles, which are the unwanted morphology. And we can see over time it converts to form 3. So these were all taken offline by a microscope. And these were then taken offline measurements for Raman. So there's a change in the Raman spectra between 200 and 400 wave number, going from a singlet for form 2 to a doublet for form 3. We were able to track that using the using the crystalline particle viewer. So we initially nucleating form 2, so these needles quite already forming this quite agglomerated structure. Then we get these individual block crystals starting to appear. So this is the starting of the conversion to form 3. And then over time, the needles start to dissolve and we get larger and more blocks appearing. And then by the end, form 2 has practically disappeared and all we're left with is a majority of form 3. And then we're also able to track that using the Raman. So this was where we ran a second experiment alongside it. So starting with form 2, we have a doublet peak at 262 wave number. And then over the course of the experiment, we see a decrease in the size and intensity of the form 2 peak, and then an increase in the size of the form 3 peak, showing the conversion to the new form. So with the new crystalline, we've improved a lot of the optics, as you can see from some of the images, but we've also improved the detection. And one of the new ways we are able to analyze our crystals and get more information about particle shape and size is using AI. So initially we have our input. So this is the input image from the crystalline. It then undergoes pre-processing. So this is where it undergoes, where we lighten the image, we can sharpen it and make the crystals stand out more. It then it's looked at through a neural network where we have trained a database with 1,000 images of each crystal habit that we are including. And with this, we're able to detect the crystals that are present based on the characteristics we've defined in this database. This then goes through particle separation. So we're identifying what crystals are present, their size and their shape. And then it undergoes statistics into an output graph. And then finally, we're able to see where and what size, what identification the crystals are, what type of crystals they are, as well as some understanding of metrics. So the 5 shapes that we've included are non-convex, and this can be considered almost like an agglomerate, where we have multiple different crystals stuck together, overlapping each other in a hard overlap. Quasi-equidistant, which is the more cubic style. Needle, which is identified by its high aspect ratio. Platelets, which are thin but with a broad in one dimension. And then spherical. And then using the additional analysis in the AI, we're able to get information about the elliptical shape factor, contour parameters and area, convex hull area and convex hull parameter, and then particle size, which is the spherical equivalent diameter of the image that's detected, the crystal. So using this, we're able to detect multiple crystal habits in the same sample. So this is a carbamazepine sample where we're seeing this during the transformation from form 2 to form 3. So we have both blocks and needles present. And then using the analysis, we're able to actually see the presence of the needles, the non-convex. So this is picking up these as agglomerates. And even the cubic, so we're seeing the blocks in green. And even though in this case the needle and the block are very close together, we're getting definition between the two different shapes. In the cases of overlapping particles, so this is the nickel sulfate with needles in the colored camera. Here there's quite a lot of crystals, so they're overlapping quite a lot. But we're still able to identify examples where, shown here, there's overlapping of needles, yet we're still able to see these as individual particles. Here there's multiple, again, overlapping needles, and we're able to identify each individual needle in the sample. Here we have a group of needles which are closely bound together, and this is picked up as a non-convex or an agglomerated particle. And then finally, we have clear particles. So this is the other polymorph of the nickel sulfate that we're able to obtain. So here we have quite a lot of crystals that are clear, so you're able to see quite cleanly through them. Even some crystals which are difficult to see by eye, so the one shown here. But with the AI, we're able to actually pick up all these crystals quite nicely. Stuff that's in the background that's not fully in focus isn't picked up. So we're excluding particles that might give false particle sizes because they're not in focus. If there's particles not fully in frame, such as here, the AI does not detect this. So we're again removing potential errors in our data. And again, these more complex shapes, it's picking up as these non-convex crystals. Now I'd like to show you the advanced analysis software live. So I hope you can see my screen. So what we have here is the full advanced image analysis AI suite. So at the top in the top of the screen, we have the magnification of the camera, the pixel size, and the focus volume. We can then change our detection method to being AI pure. So this is only using the AI detection. The blob detection, which is based on a grayscale, and the AI hybrid, which is a combination of the AI and the blob together, with the AI being prioritized. And this affects the particle detection across the bottom here. What we'll do is, in order to analyze the data, we can right-click and add a time selection. And then we can expand it to include the data we want to capture. So in this experiment, we called and held at multiple times in order to get different nucleation and growth happening. Once we've drawn a area we want to analyze, you'll see it populated in left here on the average count. And then we need to select which method is best for our data. So we can scroll through images and see which analysis method is best. We can click on them and see the effect that has on the graph here. If you want to zoom in, you double click and it will pop out. So the pure blob detection is picking up this crystal on the outside. So the outside of the crystals, there's multiple small crystals. The AI is picking up as one crystal, but then the smaller crystals aren't being detected. So the AI hybrid is actually doing both. So it's detecting these small crystals as well as this crystal as a large one. So in this case, I would like to use the hybrid. I can then do some additional features to try and filter off my crystals. So you can remove crystals from detection. So if I'm not interested in, for example, any needles, I can remove those. If I'm not interested in spheres, I can remove those. Or my agglomerated quasi non-convex, I can just remove those completely. And then the data auto-corrects to include or remove these data points. I can also see what shape has been assigned. So if I click going from size to shape, I can see that the pure blob has detected this as a plate. I can remove the opacity to see now. I'd rather think that's more of a quasi-equitant, so a cubic. If I look at the hybrid, this has identified it as a cubic crystal, which is what I think it is. Then you'll see on the right here is our particle size evolution. So this is an overlay of all the data points we've collected across the top. And we can put this in regular scale or in logarithmic. So we can then see it change over time. Additionally, I can pop this out. And I can remove or add trend lines for the experiment. So in this case, we saw initial nucleation after about 1 hour. We then saw growth. So we start to see a slight shoulder appearing and then more secondary nucleation. Again, with more secondary nucleation, but additional growth. So now we're getting particles into upper 30s. More nucleation and growth. So now we're getting particles into the 50. Additional nucleation. So now we're up to 60 microns. And then it seems the secondary nucleation takes over. And by the end, we have just a large amount of small crystals where the growth really hasn't changed in the last 2 time points. So using this, we're also able to get information about the D10, D19, D50 across the experiment. You can see the sort of same trends where above, after a certain amount of time, there was no real change in the particle size, and everything seemed to be more secondary nucleation occurring as the D50 started to decrease after a bit. So in conclusion, crystallization is a commonly used process for purification, but as well controlling and influencing particle properties, and requires quite a large amount of experiments and data in order to fully understand what's going on in the experiment and in your crystallization. The Crystalline is a versatile platform, can be used for designing crystallization at milliliter scale, gaining quite a large amount of information, all non-invasively, so not an actual contact with your sample. The solubility is the foundation of a good crystallization process. And using the Crystalline's cameras and particle detection, we're able to determine the solubility when transmissivity is not as reliable or possible. Using Raman, we're able to track polymorphism, and we showed 2 examples where we're able to track the conversion of a material in different polymorphic forms. And then finally, using the camera, able to visualize the particles in solution. And then using the AI or hybrid or blob detection, we're able to get information about the particle size distribution and how it changes over the course of the experiment, and get statistics about the D10, D50, and D90, which will give us a good understanding and foundation for designing a future crystallization scale up. So I'd like to thank you for your time, and I'm happy to take any questions.
Thank you very much, Thomas. Really a nice presentation and a deep dive into the newest features of the Crystalline version 2. Nice to see also the more simple part of the solubility, where if you have a crystalline instrument and not a crystal 16, for example, and you would like to determine the solubility, or if you have colored compounds or darker type of compounds that they were always a challenge for us. Now the crystalline with its camera technology, they are it is a solution, the solution. And it's nice that you have shared all this with us. We will also have on our website soon also a tutorial on how to, for those that they have already an instrument in their lab and they want to know step by step on how to perform such an experiment. So this is good that we could share this with our customers. And of course, the biggest added value of this software is the AI, the multiple methods that for particle detection. Previously, it was hard to detect between overlapping particles. They were seen as one big particle. And really the added value now, depending on the type of particles that one has, as you have already shown, AI or the blob or the hybrid, they do a better job. Do you already have a feeling a bit on what works better for? I know that we need much more. We need more images. We need more images. We have a lot, but still, I think not enough to train also the database for the AI, but also to be able to draw some, not conclusions. Maybe it's a bit too much to say that, but at least to see some trends. Do you already have an idea, or you would be careful in going into that direction?
I mean, we understand that there's not going to be a detection method for all cases. I mean, it's kind of why we designed the software to have the user decide themselves if they want to use the hybrid AI or blob.
Yeah.
I think you can see maybe the limitation already with the blob, and it's, if I could pull up the image, if you have, say, quite clear crystals or something that's not fully in focus, and I've seen this in some samples already, it picks up the outside of the crystal.
Yeah.
But then at the same time, the AI doesn't always pick up the small ones. And again, the AI is still very new, so it's going to learn and be better at picking up those crystals. But I find if you're having a lot of small, very small crystals with very tiny particle size, then typically the blob is more favored. But then larger crystals, typically the AI is preferred.
Yeah. We do have a few questions, although a few questions are also related to the Crystal 16, to be sincere. The first question was related to the steering bars. So what size steer bars do you recommend for the vials? I find that the wells are not steering at all in the middle of the plate for Crystal 16 when I go to check them on. Okay, so they are putting the vials of the Crystal 16 on a plate, a steering plate, and then they have the feeling that they do not steer. I do not recall now. I think they are 7 millimeters long.
I believe so.
Yeah, the ones for Crystal 16 and 10, I think, for or 12 for the crystalline. It highly also depends on the viscosity, I guess.
Yes. So we actually found the stirring in the crystalline with the overhead stirrer was better for highly viscous material than the stirring in the Crystal 16. I just think that's down to different dimensions of the vial. We were able to get a very viscous sample to get better mixing in that case.
Yeah. But in this case, the question is related more to the, I think, the bottom steerer.
Yeah.
If you have a bottom steering, so a steering bar. What a lot of customers are using is the a rare earth type of steering bar that creates a stronger type of magnetic field, and it can steer better in viscous type of or fixed slurries. And if you are using the top steering, then you need to put the magnet on the top side of the vial because in the cap are the magnets. If you, Lee, if you have more questions related to this, please contact us directly. Maybe we find together something to see what works better for you. Then another, yes, one of my colleagues just confirmed, I see the package, the bag of the steering bars 7 by 2 millimeters for the Crystal 16, and the same size, but rare earth, they do, from experience, I say, a better job than the normal ones. What's the concentration of the carbamazepine in the solvent to get those needles and block crystal images? That's
to be sincere, I do not remember off the top of my head. I know we have an application note on this, and there we do have the concentrations.
Yeah, indeed. On our website, we have an application note with Raman with Tornado, yes, where
yes.
We have the information there. Francis, if you do not find you are not able to find the application note on our website, we will send it to you directly. Then I have another one. Thank you, nice presentation. How do I see the particle aspect ratio from image analysis window?
I would probably wouldn't say there's a direct way of getting aspect ratio. Maybe the closest we can get here is the elliptical shape factor. So sort of how, I believe that's related to the height and width of the largest and the shortest side of the particle. But that's not I would say, a one-to-one relationship to aspect ratio.
Okay. Other questions. Does the crystalline measure quartz length or particle size?
So we are able to get particle size. So as I said, it's the circular equivalent diameter. So each pixel is calibrated for a certain size, so many microns, and the software will pick how many microns are inside that crystal. It then gives you the equivalent circle diameter to give you the particle size in that case. Chord length is different because it's the length cutting through a particle using the laser in the FBRM
technology. Yeah.
It's not a one-for-one, but I believe there is work within universities to do a mathematical transformation to look at that, but I cannot say if that's possible at the moment. But at the moment, we are unable to do chord length within the crystalline.
Okay. Another question related to the aspect ratio. I think you have just answered it before. Another one, oh, interesting. Does solvent refractive index influence the camera capacity?
No. So because we're measuring just the image and what's being taken by the camera, there's no real change in the in what's being seen. The aspect ratio, the solvent property usually is affecting the LED transmissivity because we're measuring how much light comes through, and that's affected by the solvent property.
And one more question, comparison between the Crystal 16 and Crystalline. Since the cloud point was detected from the number of counts, so number of particles in crystalline, so from the image analysis, it may be not as sensitive as crystal 16.
That's a valid point. Because
Thomas,
yes.
I was not finished.
Sorry.
Have you checked the solubility reproducibility between crystal 16 and crystalline? Please share with us your idea behind the two methods.
Yeah. So to answer the first point, it's a valid concern because what you have to consider is the area that's visible using each detection method. So the LED in the crystal 16 is covering the entire cross section, or a very, very large cross section of the vial. So anything that passes through that will be detected. The camera is detecting what the camera, the volume that the camera is seeing, and that might not be as large or as a proportionate area as the maybe LED sensor is detecting. I haven't done the full, one-to-one comparison for crystal.
We do have, Thomas. We do have, and actually, we have seen it in several experiments that the cameras pick much sooner, faster, the very first crystals, or the very first. Well, actually, the cloud points. There is the question mark always, much faster than the transmissivity technologies of the LED. Also because actually, of course, it depends on the resolution of the magnification of your camera. So you see a certain square, let's say, of depending on what you have, and that is actually a much larger than the LED beam. So is the other way around, Thomas. So actually, you see much faster, the chance of seeing faster the very first particle, it's higher. But there, it is more complicated than it seems. Also, the diameter of the crystalline vial is also larger, much larger than the one of the crystal 16. And I think it was in the group of Jopter Horse some long time ago, where they looked at the two crystal 16 versus the crystalline, and they concluded at that time that really the crystal 16 has the ideal length path from source to detector with the diameter of the vial, that it makes it so accurate. Nevertheless, if I have to choose the most accurate method for the crystalline, for example, I would go myself, but I hope you also agree with me, I would go with the images, with the number of particles, because the image will pick up faster than the transmissivity, the very first particles. It's kind of a delay of 1-2 seconds or so, I think, between them. So if you want to be accurate with the crystalline, I would go with the images. But also it depends on your objective, what you are doing, because crystal 16 is our instrument for solubility. It helps you in one run, 4 samples, 4 concentrations, fast, easy to do it. With the crystalline, you need to take in account that you have 4 independent reactors, and so you need. It's not that more complicated, but it's not as simple, and as you also need to use more material, and so it highly depends on the objectives that one has for this solubility determination. So there are pros and cons, but if you have a crystalline, I would say make use of the cameras for determining the clear and cloud point is the most accurate, and if you have a crystal 16, definitely you are on the right track with solubility determination. I think, let me see, Thomas. We will take one more 2 questions. If necessary, do you train neural networks for the image analysis for customers?
That would think is at a very early stage. Obviously, coming soon, we will have an annotation tool which allows users to draw and train their own databases or use our database to add additional images to it. I'm not sure how the training of the database. I don't know if the decision has been made how exactly that's going to proceed.
Another question related to the Raman. Usually, the Raman detector is light sensitive. Has there be any considerations when setting up the crystalline when the Raman probe is attached? And can the new Raman probe be ordered and attached to an older crystalline device? Yes, all the crystalline devices can be connected. Version 1 and version 2 can be connected to a Raman. We still do not do Raman and image in the same reactor, so then you do not have to be worried about the light. But there are technologies out there. Also, I know from Tornado, for example, is the latest one that we were working on, that they have camera and Raman on the same probe. So there are ways to do it. I think it's on and off, one or the other one, not both of them in the same time. So there are ways on how to, but definitely in the crystalline is not a problem. The setup, how are the different reactors setups. Does the crystalline cameras have autofocus? And if not, what happens if particles are out of focus?
It doesn't have autofocus. You have to focus them manually, but it can be quite easily done using the slider system within the settings. If they're out of focus, then they probably won't be as well detected. The main issue we see with autofocus is because there's crystals constantly moving in and out of the plane of view, the autofocus would have to move very, very fast in order to constantly keep changing that in order to maintain all the crystals in focus. It's something we might look in the future, but at the moment it's not possible.
Yeah. How does the training? But just 1 second. So it's quite easy to focus. You have a sliding bar, a few things that you can adjust, and it's quite easily to see when you get the image sharp or not. And if you really go totally wrong, you have a reset button that gets you to the beginning, and then you can do it again. So there are not that many worries related to going too wrong or going wrong or so, anything. It's pretty simple. I think we also have something on our website showing this, a tutorial on that. Somebody else is asking, for a long run, 7 to 10 days, do you have any experience of crashing out of the software? I think we have run experiments not 7, 10 days, even weeks, and we had one recently for 41 days. Anyway, it's not. I don't.
With the crystalline, it can be related to the size of the data.
Images. Yes.
The images are quite large.
Yes.
Data size. We had one instance where over the weekend we were able to fill a 1 terabyte hard drive with 7 experiments because they were collecting an image every second.
Yes.
What I would recommend is maybe reducing the number of images per second. And you can add additional steps in the software to say, for the first 5 hours, I want to collect an image every minute. And then you add another hold and say, for the next 2 hours, I want to collect an image every 5 seconds. And then add another step which says, every hour, or for this hour, I want to collect an image every second. That does kind of imply you need to know a bit about your process to try and optimize the image capture rate. But if you're doing such a long run, I would recommend reducing the how many images are taken per second. I think as standard is 1 image every 5 seconds.
Mm-hmm.
I would say reduce that down. You could do 1 image every minute if it's going for 7 days.
Yeah. So, well, the recommendation one needs to be to prioritize a bit, like where I do not need that many pictures and where I really need the information. So to take it more often, the pictures, and then if you make an estimation in that way. Usually, if you do this type of experiment, you also know more or less where you expect something to happen and where you need to look more deep, and so it's more like an optimization type of. And then a last question. Oh, I don't know if I picked everything now. How does the training for AI work? Is it made with updates? Yes, it will come with updates. The AI package is also a separate package that you can purchase while you are when you purchase the Crystalline version 2. This software only works with the version 2 and is not available on the version 1. Once you purchase it separately, you will also have access in the future to the updates, if I'm not totally wrong now. Do you have any case studies involving macromolecules, proteins? I think we do have peptides. It's a bit bigger than a small molecule. It's not a protein, it's a peptide, but we do have at least one or two. One that I can remember now on stability of the formulation of peptides on our website. We can provide it to you in an email, Joanna. I think this is it, Thomas. I think I covered all the questions. If not, we will look once again after this webinar and we will answer directly by email. If you have more questions, please write to us at info@crystallizationsystems.com or directly to one of our email addresses. If you'd like to test the Crystalline and its new features, let us know. The same, write to us or via the website. Is very easy to get in contact with us, and we could also place an instrument in your lab for some time to have the glimpse of the new software and what you can do and what this instrument can do with your compound. If you need any other information, the same, let us know. Until next time that we will see each other will be already in the new year, 2024. Until then, I wish you all happy winter holidays and stay tuned. We will come with more news and more topics to share with you. Thank you, Thomas. Bye, everyone.